Okay, no problem. Yep. Okay, thank you all. Everybody, please find your seats. We'd like to get started. We've got a lot of information that's critical. And we're going to put a lot of puzzle pieces together for you here. Well, actually, we're going to present you with all of the pieces, and you're going to put them together, because that's the way it's supposed to be. So I'd like to introduce the COVID-19 pandemic response, past, present, future analysis and lessons from frontline physicians and scientists. First, allow me to introduce Dr. Richard Urso. He studied medicine at the University of Texas Medical School, and he was part of the Honor Society, Alpha Omega Alpha. Residency in ophthalmology at the University of Texas at Dallas, a fellowship in oculoplastics and reconstructive surgery at the University of Texas at Galveston. He became an assistant professor at the Department of Ophthalmology at the University of Texas School of Medicine until 2005, also assistant professor, and became department head of head and neck surgery at MD Anderson Cooper and the chief of orbital oncology. Without further ado, Dr. Richard Urso.
Thank you. What do I push to move forward?
It just works.
It does what? Do I have to push a button to move forward?
Arrow button.
Oh, this one?
Forward arrow.
This one here?
Forward arrow. Yep. See if it pops. That's moving down. There it goes. I was using up and down.
Okay.
Up and down. Yeah.
Okay, great. Well, this is one of the funnest topics I can talk about. I was supposed to talk for about 30 minutes, so I have a lot of slides. I'll try to go through them quickly. But in general, I didn't. Craig didn't mention it, but my background, I did drug development. I actually invented an FDA-approved drug for wound healing. I got really involved in this because as I was becoming an ophthalmologist, I decided that I wanted to fix everything, and I couldn't find a way to fix everything. So I decided if I went into wound healing, that crossed all the boundaries, and I could have a lot of fun thinking about things from every different specialty and try to put them together and help to come up with good ideas. And that's basically what got me into, I did 2 years in a biochem lab and 9 years in tissue culture lab working on different projects. Primarily the tissue culture work was really innovative. I had a great mentor who taught me a lot, and Madeline Duvik taught me about how to work with skin equivalents, and I ended up coming up with there's nerve growth factor, won the Nobel Prize, but nobody could figure out how to use it. And eventually was able to use it in wound healing. I found some nerve growth factor receptors on a cell line, on an epithelial cell line, and I thought this must be here for a reason, and it turned out to be, a terrific model for closing diabetic wounds and other wounds on the body. And it's now, after 20 plus years, approved. So because of that, with that being said, I started realizing that going that route with novel drugs was always really tough, and I said, I'm gonna start looking at repurposed drugs. And this current regimen for Delta, I would call this who's who in medicines. All these things here, I call these things that I have here are almost all wonder drugs of their own. Really amazing. Ivermectin, hydroxychloroquine, azithromycin. They have so many different effects on so many different tissues, but so do a lot of these other things. And I'm going to keep myself moving. I apologize. I can't get the slide to work. So, okay, slowly. Somebody. Yeah, because I'm just pushing. It's just going down the thing. So that I kind of jumped ahead. I didn't realize it, but that's how I got involved in inflammation and wound healing, so I could cross over specialties and have a lot of thoughts to come up with ideas that might work in many specialties. One of the first things I looked at was keloids, and one of the things that I thought about was keloids don't respect boundaries. So I thought it's like a cancer cell, and if I could figure out how to slow down a keloids, I might be able to figure out something that might work for tumors. And eventually, I actually have a scarring disorder here on the eye. You see the eyeball there. It's something called the pterygium. And again, this goes back to sort of basics of wound healing. On the top of the eye is something called the conjunctival epithelium. It's like the epithelium on your skin. And then underneath is basically the dermis, what we call in ophthalmology the Tenon's capsule. So when you think about it, you think there's not an epidermis and a dermis. There actually is. And I found a lot of things that were, on this one disorder, I found a lot of interesting things about fibroblasts, and that most of the time in chronic healing, a lot of what was happening with fibroblasts. So that was a really important discovery for me as I kept looking. And then eventually, when I went to one of my wound healing meetings, this is where I got into diabetic wounds, because one of the mentors there for me basically said, hey, why don't you take your ideas and apply them towards chronic wounds? I think you can do a lot with that, and eventually that's where I came up the nerve growth factor discovery, and it works for that and other wounds on the eye also. So this is where I was saying, this group of drugs that I have on here are all drugs that I use for different things. And I would say if you had this group of drugs here, you could probably cure almost every disease. And I used them for different things: diabetic retinopathy, vascular disease, cancer, Sjogren's syndrome, fibrosis, scarring, chronic wounds. You see the list: viral disorders, etc. So I found a lot of uses for a lot of those drugs. And so when it came to COVID, what I found right away was that I know this is going to be just like every other upper respiratory viral disease, kind of biphasic. Going to be virus early and viral particles late. And for some reason, I felt like the only person who seemed to know that. I don't know why. Every time I talked to people about it, I was like, well, it's going to be an infectious disease early, but it's going to be an inflammatory disease later. And here's why I knew it. I'm going to show you this disease. So Dr. Held is here. She knows what this is. Basically, this is a picture of an eye that basically had a viral conjunctivitis. And so we know that this causes about 5 to 7 days, you're going to have a virus there, then you're non-infectious after that. And this is what we call the cytokine storm in the eye. This comes on about day 8, 9, 10, 11. And so I knew early on in February when I was hearing about viral disease and this inflammatory phase, followed by also the thrombotic phase, which is another riddle, basically. It's still a riddle that I still don't know the answer to. And I'm sure I'd love to hear, Dr. Malone has a lot of thoughts on it, so does Dr. McCullough. But basically, it was clear to me that this was going to be an inflammatory disease and that you had to use steroids early on. So my first patient back on March 10th or 12th, I used steroids, and it was about day 9, and I took a lot of criticism for it, but it was a friend of mine from medical school, so he trusted me, and we used them, and it worked quite well. But I also used hydroxychloroquine, azithromycin, vitamin D, and aspirin. So if you look at this, this is showing that the virus is rarely detectable beyond 8 days. That's SARS-CoV-2. So SARS-CoV-2, we're not finding this virus past day 8. So can anybody here tell me why we're using remdesivir? I mean, does that make any sense? How does the ophthalmologist know that and the ICU people don't? I have no idea. So really, I mean, that's a silly thing. I really thought that this was one of the things that, I won't say who, but I talked to people even when I was in Washington from the NIH, and a lot of them didn't know that these viruses are gone in just a few days. And there are still patients dying somewhere getting on remdesivir, but has no efficacy at the late stages. So this is the other riddle. I just had a talk with Dr. Malone, and he enlightened me on a lot of things. But I was really puzzled by this. I'm sure everyone here was. Why were we getting the thrombosis? Why? It was so hard to figure out. And I thought I had it down. I started thinking about fibrosis and what cells create fibrosis, and I thought about pericytes. So their mesenchymal cell line of fibroblasts and pericytes probably from the same cell line. So I thought, I bet the disease that's where the disease is occurring. Of course, they surround the capillaries, they surround the outside of the vessels, particularly the veins, and they could be responsible. Dr. Malone gives a great explanation for it. Maybe he'll enlighten us later, but how that all works. But basically, I was really concentrating on that. And so I thought maybe they're from the same cell line. And at the time, I thought maybe losartan, I had been using that as an antifibrotic agent. And so I thought, I wonder if that'll help in SARS-CoV-2 and has effects on angiotensin 1 through 7. And then you're going to basically, if the virus hits the ACE2 receptor, it downregulates it. And this creates increased vascular permeability when the ACE2 receptor is downregulated. So you use an angiotensin receptor blocker to basically help upregulate the ACE2 receptor and keep the fibrosis from happening. So I was all excited about it, and I started using it back in March. But I kind of abandoned its use because a couple of papers came out, this thing shown as an endothelial disease. And so I was like, well, maybe I'm just beating down the wrong horse. So it's such a riddle that I think we have to always use the literature to help us as we prepare and we move forward. But again, there's always a riddle, like what is really happening. I've come to think that that it's still back in the pericytes, and I'll tell you why I kind of think that's true. If you see the myocardium is being affected significantly in the pericytes in these vaccinated patients, I think it's because the lipid nanoparticles can kind of help it get through real easily and get to the outside of the vessels and cause this problem right here. So I'm not sure about it, but I think that's a good explanation for why the myocardium is attacked so much, because it's affecting the pericytes. The other thing about the issues with the thrombosis and the neutrophil extracellular traps, this is a really important thing that's happening, and you're going to see a rising D-dimer. And it surprises me how many people don't know about this in the ICUs, that there's such a thing as neutrophil extracellular traps. We know this when we do patients with cancer. They have a lot of coagulopathies, and this is a very important type of coagulopathy you see in cancer patients. And here you see the cytokine release that occurs. You see the neutrophils trapped in the platelets and creating a thrombus. And this is an important component of what's happening in this disease. So now I'm changing gears. This is my favorite drug for the last probably 30 years. I've used it for dry eye and other things, viruses also. Hydroxychloroquine. And I think one of the things that got me excited was thinking about how it works. The endosomal acidification process is a great strategy. It covers a lot of different viruses. And I always say that my favorite thing to say is, when I tell my patients, hey, I'm going to put you on hydroxychloroquine, and they say, what are the side effects? And I go, it lowers the glucose, lowers the cholesterol, lowers the hemoglobin A1C, lowers the inflammatory markers, D-dimer, sed-rate, C-reactive protein, lowers the risk for, lowers atherosclerosis and accelerated atherosclerosis, helps in lower stroke, heart attack, and pulmonary embolus, decreases chronic kidney disease. There are now 96 clinical trials going on in clinicaltrials.gov for the usage of hydroxychloroquine in cancer. So I say, those are your side effects. So. This study was an aha moment for me. This came out, it was actually, the preprint came out on March 10th. My first patient I treated was March 12th. And I was so excited when I saw this. I was like, oh, I have an optimization strategy. So I thought that everybody was going to adopt this. And there was some real, right after this, Didier Raoult came out with some great information that hydroxychloroquine helped. And so I thought, this is going to be it. We're going to get this disease under control. We're not going to let patients get on vents and ECMO, and this is going to be it. We won't even need the 2 weeks. I remember thinking about that. I told my office one day, I said, I had a couple extra hours. I said, I jokingly said, I'm going to go end this pandemic. It was before we ever had a lockdown. I was joking, but I was partially serious, but thinking back now, it's pretty naive. So there's some other wonderful things. You saw some molecular modeling studies of hydroxychloroquine that showed that it helped, and this is Fantini. He did this in silico modeling studies that were wonderfully done. I always thought, and I'll still say this to this day, hydroxychloroquine, I think in low doses should be used late in the disease. They've said it doesn't work, but it's helping, I didn't say this, but it helps platelet aggregation, it helps neutrophil extracellular traps, It's doing other things chronically, of course, for inflammation, toll-like receptors. It should be used at low doses late in the disease. I think it's a real mistake not to use it. I hear it over and over again. I think the literature says otherwise. Reduces extracellular traps. Okay, so the other drug that I've actually used on the eye for about 16 years is ivermectin. So we use it for mites, those little mites, Demodex mites that get on your eye, but I also use it for viral disorders, for DNA viruses. I thought it had some effects, so we'd use it sometimes as an inexpensive way to help with some herpes virus. Also used it orally whenever it had cytomegalovirus or Epstein-Barr virus. So I've been using it for many years. But I couldn't figure out how it was working late in the disease. So there's a clinical overview that was put out. It's got 20 mechanisms. But basically, one of the things that I was trying to do early was figure out how is it working late. So John Jacques Ratcher had done some studies, and he showed that in the ICUs it was working. And I really couldn't figure out. I knew it had these antiviral actions. I knew it had some effects on lipopolysaccharide. That was well known. But it also has a whole bunch of other effects. And you'll see in this paper, if you have a chance to read it, I won't talk because I don't have enough time, but it's got a lot of different effects, anti-inflammatory. It's antithrombotic, so it's affecting CD147. It's affecting, well, you see NF-kappa beta. And this also affecting neutrophil extracellular traps, activating PTEN demethylation, and also lowering myeloperoxidase. It's actually helping neutrophil extracellular traps. So it has a lot of different mechanisms as an anti-inflammatory. So when you think about ivermectin, you think about hydroxychloroquine. Don't think of them as just attacking the virus. They actually affect the thrombotic side of the disease, and they affect so they're affecting coagulopathy, and they're affecting the inflammation in a big way. So this is, I'll have to end the talk a little bit early, but this is another, what I would consider to be a wonder drug. This drug has basically stopped for me, on the surface of the eye. I have a scarring disorder I showed you earlier, the pterygiums. I've been using this over 20 years. And for a long time, I knew it affected the modulation of fibroblasts. But for a long time, I just thought I was a really great surgeon, you know. But actually, when I started realizing I was never getting any recurrences, I was using this for 4 months along with steroids. So steroids are number one, but this was the number 2 drug. And I really felt like it had a huge impact on my practice over the years. It's a really common thing when I showed you that pterygium that comes. And it literally, it's a fibroblast disease, and this has a massive impact on the fibroblasts. It affects all these viruses. And you see all the different, if you have a chance, you see all this written here, the immunomodulatory effects. This is a great paper right here, the immunomodulatory effects of macrolides. I usually tell people this is a better immunomodulatory than it is an antibiotic. It's amazing. So I would really try to put it to use in your practice. I think some people know in chronic pulmonary issues, it's useful using for chronic, maybe for 30 days or during the wintertime, 60 days at a time. It's a great drug, and I would definitely move forward with it. And here you see an outline showing all the effects on neutrophils, eosinophils, monocytes, T cells, macrophages, and fibroblasts. So it's affecting at multiple levels the immune system. So I'm going to take up too much time. Dr. Malone, you might want to ask him about this question about famotidine. He's running a clinical trial, so I won't go over that because he's an expert in that area right now. I want to just go over this. This is my new favorite drug for SARS-CoV-2. Actually, when it came down to Delta, we started having, I've treated about 1,000 patients, by the way. So I've treated about 4 today, actually. I did on telemed. But what I was going to say, cyproheptadine made a huge impact on my practice. And I got the ideas from various other people, just like everything. Almost none of these ideas are my ideas. It's just about putting the riddle together, putting the pieces together. Dr. Shetty from South Africa, who's treated like 6,000 patients, talks a lot about attacking the mast cells. And so it's a great H1 receptor antagonist, and it also is a great serotonin drug. So there may be a role for serotonins. As you know, the megalokaryocytes have a lot of serotonin in them, and they're in the lungs. The degranulation of those may be causing some of the problems. I don't know if anybody's read that kind of work, but that's been there. And this is a great drug to block serotonins. It's also anticholinergic. And I think, for the most part, I think, you see I have other drugs here. Singulair, you all know about Singulair. Fenofibrate, this, again, okay, here's another one of my super favorite drugs. I've used this for tumors, for neuroectodermal tumors. My patient with melanoma, well, it was the first time I came to my mind, it was for glioblastomas. But it has all these other wonderful effects, anti-inflammatory, antithrombotic, and as I said, antiviral also, and anti-tumor. So I think you ought to put this in your regimen of drugs that you use more frequently. And it showed that it significantly decreased the SARS-CoV-2 in cell culture models. These are all done in viral kidney cells because viral kidney cells don't fight back. And it was a 70% reduction in these cells. So I think I'm going to go ahead and end it there. For people, well, let me just cover this. Dutasteride, for people who haven't used it, I started using it about 3 or 4 weeks ago, and it works really well. Early in the disease, it decreases the viral shedding. And by decrease the viral response, and by doing that, it decreases the inflammatory response. And here's the current regimen, as I said again. We're always going to be faced with new and untreatable disorders, and often the answers are right in our toolbox. That toolbox, I said, is there's a lot of things you can cure right now in your office that you're not even thinking about that you can do. Well, thank you.
Thank you, Dr. Urso, for an enlightening presentation. Please save your questions till the end. We're going to save some time for questions. Our next speaker is Dr. Ryan Cole. He attended Medical College of Virginia for his MD degree. He did a year of PhD research in immunology. He did residency in anatomic and clinical pathology at the Mayo Clinic. He did surgical pathology fellowship and was the chief fellow at the Mayo Clinic. He did dermatopathology fellowship at Columbia, and he was the chief fellow there. I see a trend. He is now the CEO and medical director of Cole Diagnostics in Boise, Idaho. Welcome, Dr. Cole.
Thank you, Craig. Scoot that one down. Mess up the yin-yang of the universe here. Alrighty. Got a lot to cover, so like Dr. Urso, I will try to be quick here. Okay, conflicts, I just want to share. I have a small interest in a rapid antigen test, and my opinions today are my own. We're off to the races here. Somebody on the one side of that slide may have won the race, and I don't know what drug he took, but Joe Rogan won the race. So here we go. I want to start first with a declaration. Dr. Malone, Dr. Urso, and I were lucky enough recently to participate in drafting a resolution to the world, basically. And I think it will resonate with the people here, and we invite you to opine and perhaps sign on with us. We, the physicians of the world, united and loyal to the Hippocratic Oath, recognizing the profession of medicine as we know it is at a crossroad, are compelled to declare the following. Whereas, it is our utmost responsibility and duty to uphold and restore the dignity, integrity, art, and science of medicine. Whereas there is an unprecedented assault on our ability to care for our patients. Whereas public policymakers have chosen to force a one-size-fits-all treatment strategy resulting in needless illness and death rather than upholding fundamental concepts of the individualized, personalized approach to patient care, which has proven to be safe and more effective. Whereas physicians and other healthcare providers working on the front lines, utilizing their knowledge of epidemiology, pathophysiology, and pharmacology, are often first to identify new potentially life-saving treatments, whereas physicians are increasingly being discouraged from engaging in open professional discourse and the exchange of ideas about new and emerging diseases, not only endangering the essence of the medical profession, but more importantly, more tragically, the lives of our patients. Whereas thousands of physicians are being prevented from providing treatment to their patients as a result of barriers put up by pharmacies, hospitals, and public health agencies, rendering the vast majority of healthcare providers helpless to protect their patients in the face of disease, physicians are now advising their patients to simply go home, allowing the virus to replicate and return when their disease worsens, resulting in hundreds of thousands of unnecessary patient deaths due to failure to treat. Whereas this is not medicine, this is not care, these policies may actually constitute crimes against humanity. Now, therefore, it is resolved. Thank you. Now there it is resolved that the physician-patient relationship must be restored. The very heart of medicine is this relationship, which allows physicians to best understand their patients and their illnesses, to formulate treatments that give the best chance for success while the patient is an active participant in their care. Resolved that the political intrusion into the practice of medicine and the physician-patient relationship must end. Physicians and all healthcare providers must be free to practice the art and the science of medicine without fear of retribution, censorship, slander, or disciplinary action, including possible loss of licensure and hospital privileges, loss of insurance contracts, and interference from government entities and organizations, which further prevent us from caring for patients in need. More than ever, the right and the ability to exchange objective scientific findings which further our understanding of disease must be protected. Resolve that physicians must defend their right to prescribe treatment, observing the tenet, first do no harm. Physicians shall not be restricted from prescribing safe and effective treatments. These restrictions continue to cause unnecessary sickness and death. The rights of patients, after being fully informed about the risks and benefits of each option, must be restored to receive those treatments. Resolve that we invite physicians of the world and all healthcare providers to join us in this noble cause as we endeavor to restore trust, integrity, and professionalism to the practice of medicine. Resolve that we invite the scientists of the world who are skilled in biomedical research and uphold the highest ethical and moral standards to insist on their ability to conduct and publish objective empirical research without fear of reprisal upon their careers, reputations, and livelihoods. Resolve that we invite patients who believe in the importance of the physician-patient relationship and the ability to be active participants in their care to demand access to science-based medical care, in witness whereof. And this is at globalcovidsummit.org. And then the declaration, if you would like to add your imprimatur thereon, is at Doctors and Scientists. It's at the bottom of the globalcovidsummit.org page. And this is where we are as of last night. We have 7,800 doctors and scientists who have joined us. All right, one of my favorite quotes, if everybody is thinking alike, then somebody isn't thinking. General George S. Patton, and that's where we are. Here's some points to ponder from a pathologist's perspective in 10 plus posits in 10 minutes. Ready, set, go. SARS-CoV-2, cultures. Okay, some people, is it culturable or not? Well, it looks like it probably is. And there's some articles, start with that one, culture-based isolation to evaluate infectivity, viral cultures, et cetera. Now I know I love Dr. Merritt and we're best buds, but at the same time, I think there are some articles that show we can do it. Now here's kind of the logic argument. In the ivermectin trials in Monash University, if you look at that and we say it inhibited viral growth, in order to say it inhibited viral growth, we had to culture it. So that's just a logic game to play with that one. All right, testing. I do run a medical laboratory, so here's the question and answers. All right, you can see on the left-hand side, proper technique. You have more ACE2 receptors, TMPRSS2, underneath the middle turbinate bone further back. On the right, you can see them trying to puncture the plate of the brain, doing it absolutely incorrectly. Okay, so with a PCR, what are we detecting? We're not even detecting the CAR on your left there, or the virus. We're detecting the blueprint for the CAR, okay? But we're not even detecting that much of it. We're really looking for, and this is thanks to Dr. Urso, we're detecting the CAR parts, the blueprint for the CAR parts. So PCR is only looking at a part and a specific, hopefully conserved sequence of a virus. PCR, with RNA virus, you have to do a primer, turn it into DNA, and then amplify. And then we amplify it, you hear your CT values, and people complain, well, what does it mean, what doesn't it mean? I'll tell you what it means. Here we go. Is a PCR meaningful or meaningless? Well, these tests should be used in symptomatic patients. If you get a CT of 34, it just means something's present, the blueprint for a car part. If you get a CT of 15, you have a lot of blueprints for a car part. But what if we were doing it this way? Patient has a CT of 34 and is starting to become symptomatic, and 2 or 3 days later now has a CT of 15. Well, they're going into disease. PCR is semi-quantitative. In that case, it's meaningful. A snapshot in time means nothing. Patient has a CT value of 15. The number of times you amplify, now a week later or 3 days later, they're at 34. What are they doing? Going out of viral replication. Patient comes in at a CT of 34, and a week later is still at a CT of 34. Well, they still have blueprints for car parts. And we're not looking at it this way. So using a one-time test and saying, you're a case, is insane. We have a case-demic because we're misusing a test that can be a good test when applied in the proper way. What about antigen tests? Okay, now we're actually looking at the proteins. We're looking at the car parts themselves, not the blueprints for the parts. In these lateral flow tests, these quick antigen tests, we have anti-antigen antibodies bound to a nitrocellulose. We have reagents that will glow those. So we're able to look at those and say, okay, protein present, protein not present. Those, again, are good in context. Symptomatic patient, there's a lot of false positives with all of these tests if you run the cycles too high on the PCR and if you test asymptomatic patients with these tests. So using a test the proper way at the proper time is what we should be doing, and we've gone crazy societally overusing testing. And that's from a guy that runs a lab. You know, I don't mind going back to just reading biopsies and not doing way too many PCR tests, I promise. Antibody testing, there's lateral flow tests, there's some good ones, there's some bad ones, a lot have been pulled from the market. In the laboratory on the right, we actually use big machines and chemiluminescent tests. T-cell memory tests, there's a good one out there, the T-Detect, usually about 15 days after the infection, you can start detecting the genes that are turned on and turned off in the virus. 95% sensitive, 99% specific. There's others coming onto the market. That's simply a blood draw, and you can send those off. All right, where are we now? Delta variant, is it real? Yes, it is. It's more transmissible, certainly. There are new mutations in it, multiple at the receptor binding domain, a lot of substitutions that make it more virulent. This is the one that really makes it interesting because at the furin cleavage site of the delta spike, it splits more easily. 10% of bound virus would get into cells previously. Now with delta, about 75% of bound virus because of this mutation can get into cells. So why do we see delta going like a wildfire in patients? That's why these new mutations have allowed it to do so. All right, question, pathologist, where are all the autopsies? And Dr. Merritt brought this up earlier. That's a really good question. And we've had a novel new disease, supposedly. Why in the world don't we have billions of funding looking at every single death and documenting all the changes that we would expect or maybe not expect? We can't do science if, where are the billions? Where are the autopsies? Crickets. One cannot find that for which they do not look. That's plain and simple. And if we're not looking, we won't find and we won't advance the science. So it's very frustrating. Where are the post-vaccine autopsies? Same question. I was just on the phone with some doctors out of Germany the other day, and we're trying to put some extra studies together, a doctor out of Sweden as well. Thank you. And it needs to happen. It needs to happen. After I gave a talk in Texas, I have more autopsy tissues coming my way. We're going to stain them for spike, nucleocapsid, antibody deposition, clotting, et cetera, all sorts of things we're looking for. Because in the few that we have seen in some of the mammal models, on the spike itself we know can be a toxin. It induces disease alone without the virus being present, and that's important. I've shown this slide before in another talk. On the left, blown apart mitochondria. In the lungs, on your right there, tons of inflammation in the lungs, spike alone. In the heart, inflammatory cells. The blue is early scarring. Liver, around those vessels on the left, all the blue. See, I get to look at all these pretty colors all day long. I kind of like what I do. What about the ovary? Where are the studies on the ovary? We know the lipid nanoparticles accumulate there. And then, today we have the CDC saying, oh yeah, just give the shots to pregnant women and young children and everybody. And we don't have the long-term studies for something that is experimental. We don't have 5 to 10 years of safety data on these things. So what about vaccine safety? Good question. You know, in the US, 15,000 deaths, 726,000 adverse reactions reported. That's quite a lot, even more in Europe. Up top, you can see that's 30 years worth of vaccine adverse event reporting data. And at the far right of that graph, you can see what we've seen in just a short 8-9 months. On the left, that's the number of deaths within day 1, day 2, day 3, et cetera. So you can see a proximity and an association with death right after the shot. More deaths from this shot in 9 months than we have seen in 30 years combined, of all vaccines combined. Screen before vaccine, this is something that we don't hear enough. If you've had COVID, you do not need a shot. You have natural immunity. And why does this matter? Well, you know, I know Dr. Malone will probably talk about high zone tolerance and other things, but if we are not screening the patient and they're cooking virus at the time they get the shot, you are just setting up a perfect cascade for clotting, abnormalities, et cetera. If somebody's had COVID, like lots of the children have, half the children in some studies around the world, or around the US, have had COVID. Meanwhile, we're mandating something and we're putting people at risk. You must know whether or not they've had COVID before you give them a shot, if you choose to do that. Immune modulations post-shot, what are we looking at? Pre- and post-shot labs, good idea if someone's forced by their employer to do it. If they want to document and have recourse against an employer, do a pre-panel. Do a D-dimer, do a sed rate, do a C-reactive protein, do a troponin, do a CBC, CMP, maybe add another creative test or two in there. And then after their shot, if they're not feeling too well, you might as well check those again. Now you have documentation. And the patients that complain about post-vaccine injury, check these anyway. I know a lot of our colleagues are ignoring basic science, and I would encourage them not to. Okay, what about what's happening with the shots in terms of immune modulation? This was a paper out of the Netherlands. The Pfizer vaccine induces complex functional reprogramming of innate immune responses, which should be considered in the development and use of this new class of vaccines. Well, what does that mean? Well, it means we're seeing dysregulation, misregulation, and downregulation of a transmembrane receptor called toll-like receptors, number 3, number 4, number 7, number 8. We're seeing latent viruses pop up, herpes family, zoster, HSV1, 2, HHV number 4, Epstein-Barr, RSV, HPV, et cetera. These toll-like receptors keep viruses in check when they're at normal levels. Well, post-Pfizer in this study, it's like all of a sudden, these receptors are dysregulated. Why are we seeing viruses? Because those, after the shot, are downregulated. How long do they stay downregulated? I guess we'll find out. Are we doing the science is the question. Really critical is cancer. What do you need to allow cancer to proliferate? Downregulation chronically of TL4 and TL3. So what does the shot do? Downregulation. So we're seeing more zoster. What about P53, the guardian of the genome? P53, tumor suppressor gene. Well, interestingly, studies show that the spike subunit binds to P53 receptor. It also binds to the BRCA gene. So what are the long-term implications of P53 and giving a shot that is the spike protein? That's a good question. This is what we're doing. What about interleukin-6? Chronic elevation of interleukin-6 leads to chronic inflammation, increased cancer. What about interferon-1, JAK, STAT? All these cancer pathways are upregulated. So these are really critical, important questions. All right, where'd the flu go? All right, the secret social lives of viruses. Everybody's asked this question, and this is really a fascinating area. Here's their social life, hanging out at the bar, these little viruses and whatnot. In the social life of viruses, there's something called a viral arbitrium. They send little peptides back and forth, signaling peptides. And if you look at the graphs for about 50 years in Europe and the Western countries, you can see strong cold season, low flu season, strong flu season, low cold season, back and forth. And you can see these variations. What stopped the 2009 flu outbreak was a rhinovirus outbreak. So they actually signal to each other. It's just not all the testing wasn't happening or we weren't looking for flu. I think the amount of flu testing went down, certainly, and the flu was certainly still here. But also this is a newer area in lab medicine, virology research to understand is viruses can suppress each other and talk to each other. All right, what about antibody-dependent enhancement? Are we seeing it? Probably. We were warned about it last year. We've known with coronaviruses and trying to vaccinate. We know the history of SARS-CoV-1 and MERS. It happens. This family of viruses constantly mutates fast enough to stay ahead of playing whack-a-mole with a booster shot. And because of that, we are still putting a population at great risk with no long-term safety knowledge as to whether or not this is going to happen. We know the viral loads are up. Are we starting to see binding non-neutralizing antibodies? You bet we are. What about natural immunity COVID recovered? Broad, natural, long-lasting, durable, as Dr. McCullough right here says, far stronger than a vaccine immunity. Common sense. What is a vaccine? It is mimicking a portion of a natural infection. What are we doing with a vaccine? Trying to mimic nature. I think nature has it right from the word go. And those who are COVID recovered have this fantastic antibody in their eyes and their tears and their mouth called IgA, which we don't get from the vaccines. Why are the vaccinated carrying high viral loads? They're not neutralizing with IgA like the COVID recovered are. Long-term immunity since 1918 in elderly people from the 1918 flu pandemic. Grandma's measles, she's still immune. Hepatitis A gives a rise to natural active immune response leading to lifelong protection. Why are we ignoring nature? And the other thing too, people panic about antibodies. Don't worry about your antibodies. They'll go down, but you know what goes up? Your T cell response goes up, and in SARS-CoV-1, 18 years later, the individuals that have been studied still have long-term immunity to SARS-CoV-1 from their T cells 18 years later. All right, think of the virus as a bicycle, front tire's the spike, you get a shot, you get an antibody to the spike, and a bunch of antibodies to the spike, you get an infection, you get antibodies to the whole bike and T-cell memory to the whole bike. Okay, there's the delta crash bent spike up front there. Your body, if you've had COVID, remembers the rest of the bicycle and will forever, hopefully. And to vaccinate the COVID recovered is scientifically incoherent. Treatments, thank you. Dr. Urso went over treatments. Treat early, do not wait. Delta, 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 it duplicates very quickly. COVID's a clotting disease, clotting disease, clotting disease. Don't forget the aspirin. Don't forget all the treatments that Dr. Urso did mention. I've got them all up here too, and the clock is against me. And lots of treatments. So the NIH telling us there's no treatments is insane. Don't forget to check your vitamin D levels. Vitamin D levels play a critical role as to whether you're going to get COVID and the degree of severity you will or won't have with it. And there's a study from 191,000 patients, your acquisition on the left versus your vitamin D level going to the right. Look at your comorbidities, look at your diet, cut the carbs. Patients should be on a keto diet when they're sick. Sleep affects the stickiness of your immune cells, how they talk to each other, how they function. And don't forget to defend your colleagues. There's some tall blades of grass in this room, and we're happy to speak out. Misinformation, disinformation, no, there's information. The cream should always rise to the top, but to be labeled by the brown shirts as just giving disinformation is a propaganda strategy. What we should have is just information and dialogue, and the truth will always prevail. So we don't want to be the tall blades of grass. We're happy to do it, but at the same time, we need you at our side, so we appreciate it. And at that, I will leave it until we get the questions. How did I do? Did I make the clock?
Do you mind having a seat? All right, thank you for an excellent presentation. Now I have the pleasure of introducing Dr. Robert Malone, who got his bachelor's in biochemistry at UC Davis. He got a master's in biology from UC San Diego. He got his MD from Northwestern University School of Medicine. He did a medical pathology internship at UC Davis School of Medicine and did fellowship research and postdoc studies in Global Clinical Scholars Program honors at Harvard. Dr. Malone has become known as one of the inventors of mRNA and DNA strategies and a worldwide expert on mRNA and DNA technologies and its relevance in biodefense and outbreaks. And now, Dr. Malone. Thank you.
There we go.
Before, actually, if you can use the space bar, though.
I would like to use the space bar. Okay. Hi, I'm Robert Malone. I'm a physician scientist, and I am also clearly labeled as a disinformation specialist. because I don't know anything about vaccines and biodefense. This is, I don't know, number 5 or 6 outbreak for me. I'm kind of an outbreak junkie. I work closely with the Defense Threat Reduction Agency. I am a vaccinologist. This is my moment of seeking absolution. You know, this is my disclosure. And I was very involved in bringing forth just one example, the Ebola vaccine during the West Africa outbreak. I was actually the guy that got Merck involved in that on behalf of Defense Threat Reduction Agency. So this is the obligatory what are your bona fides slide. I think we already had enough of that. I'd like to, and I very much appreciate that you already read the declaration, so I don't have to do it. I'm kind of coming hot off of a little bit of an international tour, which included the International COVID Summit in Rome. And this brought together physicians and scientists from all over the world, both in person and virtually. Just to give you a little taste of that, it's available online for streaming, and Dr. Cole has already identified the site where a lot of the clips are being rolled out, as well as the declaration, that being globalcovidsummit.org. Please visit. I hope all of you consider signing the declaration. It's part of a very targeted strategy. So my talk is not going to be so science-rich. It's going to be a little bit more public policy-rich. I defer to Dr. Cole and Dr. Urso, who have done a fantastic job. This is a brief summary slide of what went on during that 3-day meeting, in which we met on day 1 as a group and discussed treatment protocols. On day 2, we actually gave testimony in the Senate in Rome. It's on the bucket list if you ever get a chance to do that, worth doing. Then day 3 was a large public session. On day 2 and 3, just to give you a sense of things, those were both streamed live and through a distributed network all across the world. We had over 100 million connections on both days, okay? So a lot of interest in Brazil, Latin America, Africa, all over the place. While I was there, I also met with Cardinal Turkson in the Vatican to discuss the strategy that I'll mention in a moment. Let's see. And I don't have to read the physician's declaration because it's already been read, and so thank you for that, but I hope that you will consider joining us. Just to say something about that, this new globalcovidsummit.org site is part of a very conscious strategy. As you know, we're up against an amazingly integrated, well-financed juggernaut it's disinformation. And it truly is disinformation that we're up against. It is carefully coordinated, it's propaganda, it's backed by swarms of fact checkers that have no training in biology. Many of them don't even have a bachelor's degree. They're not trained in journalism, and they're hired by organizations that have been set up that are funded by Zuckerberg Chan, Facebook, Bill and Melinda Gates Foundation, Robert Wood Johnson Foundation, who's one of the major shareholders of J&J, if not the largest shareholder, good to know. Follow the money, okay? And these organizations, if you scroll through, many of them specifically say, hey, if you want a hit job on somebody, I'm paraphrasing, call us up, fund us. That's what they do, okay? This is not about checking facts. This is about reinforcing the dominant narrative. I can tell you from personal experience, and the thing that has amazed me in traveling through Europe over the last month is it's not just the docs here that are getting hit with this same script. It's all over the world. They're even using the same words. I'm now part of the Dirty Dozen in Rome, in the Roman press, which is noted for its corruption. Now, they identified me as part of the Dirty Dozen, a term that was used by our POTUS, you may recall, and applied to Dr. McCullough and others, including Mr. Kennedy. as Dirty Dozen number 2. In the Italian press, they used the words, but they only identified 10 of us. It was kind of fascinating. And I was also identified as a terrorist. I'm now a terrorist because I advocated for early treatment to save patients. All of us that were participating in the conference were labeled as a bunch of crazies. And here in the American press, they're doing a push, I don't know if you saw in, I think it was MedPage, that basically they're trying to say we're all a bunch of Proud Boys, right-wing crazies. This is how propaganda works, okay? Now I deal with the intelligence community quite a bit. We are being manipulated. Our patients are being manipulated. There's no ifs, ands, or buts. And it's unprecedented, the scale and the coordination that's happening globally. I've never seen anything like this. I've been through so many pandemics. Never seen anything like the information control that we're seeing now. Carrying on. See if I can make the computer work. You know, I'm not a millennial, so forgive me. I just have raised some. Yes, so in terms of my bona fides, yes, in fact, I did make these discoveries between 1987 and 1989, and there was a series of patents issued. I am an inventor on those patents. The fact checkers all, they've gone, it's fascinating over the last couple of months. Oh my good heavens, look who has arrived. I'm in awe. I really am. What an amazing job he has done. I think a round of applause for Peter. The man is a monster. He's a powerhouse of information. He knows the literature up one side and down the other. I'm worn out just watching him. So what an honor. But yeah, so the fact checkers have decided that I'm not an inventor of RNA vaccines, even though I'm on 9 different patents and I have all the patent disclosures and all the original data. So that's, rather than belabor this point, just this is the team from the Salk Institute back in the day. That's Inder Verma there. Just to let you know, it's Mark Kindy who signed off on the invention disclosure in 1988, January. This is me laying out the whole technology profile at Vical. This is the team that I worked at Vical, and this is my team at UC Davis that carried on and was responsible for the fundamental patents on mucosal polynucleotide vaccination to elicit IgA. Yay. What a concept. So, getting into the politics a little bit more. At public policy, this is a series of positions that I've worked up with Peter Navarro. You may recall Peter was in the Trump administration. And I've had the pleasure of interacting with him. We put out 2 op-eds. The first one in the Washington Times was, of course, fact-checked by Facebook, and so the Washington Times got quite upset about that. I spent a day writing the retort and documenting all the things, and they sent it in to the fact checkers and Facebook basically said, we don't care what you say. We don't care what facts you document. You're still on the blacklist. So that's how that works with these guys. This is not about facts. This is about the narrative. They don't care. You can document that they've got it wrong. They don't care. They won't change the story. I had the same experience with Atlantic Monthly. So our recommendations for policy are based on a 4-pronged approach. Vaccines for the elderly, morbidly obese, and other high-risk individuals. Now, which vaccine? That's a good question, and which are the ones that are most safe, and are these the best right now? And I think that is an interesting question that I don't have time to go into. I think that we need to monitor the safety data and may need to adjust that logic as new data arrives. Early interventions with supplements, we've heard great examples of those supplements. Ready access to self-diagnostics, knowing that they will be high false positive rates, and you can do secondary tests, as we did with AIDS, as we do with everything. We set the initial screening tests for high false positives, and then do confirmatory tests that are more specific. You know, I was trained in pathology a little bit, but not as well as some. Apps and other tools for self-assessment of true risk of severe disease and death. What has amazed me is how effective the press and the HHS monster has been in promoting the idea of fear. How much fear? I mean, the recent survey that something like a large fraction of the population believes that their probability, if they get infected, of going to the hospital or dying is something like 40 to 50%. This is crazy talk. We all know that except for that high-risk group, the probability of your patients going to the hospital, having severe disease, or dying is a fraction of percent, and even lower for the children. Okay, so it's all clustered in certain age groups and high-risk cohorts, and the rest of us are just getting scared to death by the news press, and that serves their interest and sells newspapers, I guess, and maybe other things. But we advocate for AAPS and other tools for self-assessment of risk. So why are we vaccinating children against COVID-19? I'm not going to belabor this. This is an already converted audience for the most part, but this is a fascinating recent paper if you haven't seen it right now, because it goes into a deep analysis of what the true morbidity and mortality is here from the VAERS system as corrected for known VAERS underreporting. And you may have seen the recent video out that is based on the data coming from CMMS, and I've known of numerous other groups, including Dr. Brian Bridle and his group, and Steve Kirsch, that are diving into the CMMS data, and it's consistent with these projections based on the inadequate VAERS system. Moving forward. I really don't like to leave people in talks without hope and without something forward-looking. And this particular couple of slides also serves my personal purpose, since I'm labeled as an anti-vaxxer. I just haven't been able to disclose. You may or may not be aware that I've been at the forefront of advancing the combination of high-dose famotidine and celecoxib as a treatment. We have 3 studies, large, randomized, adequately powered clinical trials, quite sophisticated, about to begin. One in India, one is an inpatient study here in the United States. And the other one is a sophisticated virtual outpatient trial here in the United States with multiple sites. I think we're still looking for site networks, and we're still looking for at least one more hospital for the inpatient group. And then it's also being done in parallel inpatient in India with high-dose famotidine and celecoxib. All those studies are about to start. The U.S. studies are funded by Department of Defense. Okay, so this is not a crazy marginal group that is just doing wackadoodle right-wing science. In addition, I've been supporting a company, I just haven't been able to talk about it, called Reliance and Reliance Healthcare. Reliance happens to be the largest commercial conglomerate in India, run by one of the wealthiest men in the world. And they have a fund that they're obligated from the Indian government to set up. It's like tithing of about $1 billion currently, and they're very focused on building a vaccine for the poor. There's about, something like 1 billion poor in India, and to vaccinate all those is going to require a product that has a very low price point, is very safe, because the Indian government is quite sensitized to vaccine safety consequent to prior experiences with the company Merck and the philanthropic organization Bill and Melinda Gates Foundation. So Mr. Ambani and Reliance has launched quite a while ago, about a year ago, a very rigorous development path for a traditional vaccine. I know people have talked about Novavax. I'm not here to pump it. I just want the public to be aware. I haven't been talking about my vaccine work because I've been bound through confidentiality with my client, Reliance. But that's now disclosed. I presented this a couple weeks ago at a major international vaccine conference. So this is a traditional product. It combines 2 different proteins, the receptor binding domain of spike, not the whole spike, and the nucleocapsid, a well-characterized adjuvant system composed of alum and CpG, and it has been rigorously assessed in terms of preclinical toxicology, unlike the RNA vaccines and the adenovirus vaccine. The non-clinical safety data are very encouraging. The animal studies are currently in progress, and we have clearance to begin a 3-arm dose escalation, safety, and immunogenicity study that should be starting in the next few weeks in India. So there is another product, and the Reliance is very interested in bringing that product into the United States as well as Latin America, and looking for partners on that. And I'd like to close with the noble lie. So this is something I've advanced, and it's now entered the popular jargon. I was fascinated to hear the governor of Florida the other day talking about the noble lie and how he believes strongly that the noble lie should not be used in public health. What I wanted to bring to your attention is these 5 elements of the noble lie that we've seen rolled out by Health and Human Service leadership, including Mr. Fauci. And it really underpins what we have been experiencing. The noble lie includes these 5 elements. Herd immunity is required to enable economic and social recovery. Vaccines are the only route to achieving herd immunity. The COVID-19 vaccines are perfectly safe. The COVID-19 vaccines provide durable protection and immunity. And natural immunity is short-lived, narrow, and ineffective. We all know these to be false. When I first started talking about this about 4 months ago, this is highly controversial, but it's now abundantly clear that these are all misrepresentations. Why would they do this? So this is a fascinating, particularly because of the date, 1984. This is from the Federal Register. The Federal Register basically is the logbook that captures federal policy and laws. And looking back on June 1, 1984, they published that any possible doubts, whether or not well-founded, about the safety of the vaccine cannot be allowed to exist in view of the need to assure the vaccine will continue to be used with a maximum extent consistent with the nation's public health objectives. Accordingly, because of the importance of the vaccine and of maintaining public confidence in the immunization program that depends on it, good cause exists to issue these amendments as the final rule effective immediately. So this has been federal policy since the prior century. What we are experiencing now is the federal government and the world health community has the ability to enforce this through propaganda and information control like we have never seen it before. But don't be surprised that this is happening. They've told us that this is what they do. They've been doing it for a very long time. This is not a unique, novel conspiracy. This has been the policy of our government for decades. It's just that now they have enormously powerful tools to reinforce it. And with that, I close, and thank you for your attention. I hope that was helpful.
Thank you, Dr. Malone, for another eye-opening presentation. And now, prior to questions, we have a special guest surprise. Fortunately, she was able to be with us today. I think she's going to give you some information that's going to help you put all of the pieces together of the pandemic and everybody's response to it. We have the pleasure of introducing Dr. Li-Meng. She's an MD from the 7-year program at the Xi'an, assuming I'm pronouncing that correctly. My apologies if I hadn't. Medical College, one of the top medical schools in China. She has a PhD in ophthalmology from China, from the Central Southern Medical University. She did postdoc fellowship with the WHO. She was in the H5 Reference Lab, the School of Public Health at the University of Hong Kong. Dr. Yan spent 5 years researching influenza vaccines when in January 2020, she investigated Wuhan pneumonia. At that point, consequently, she had to escape Hong Kong in April 2020 and has been in the United States as an independent virologist, immunologist, and COVID-19 expert. The very courageous Dr. Li-Meng.
Thank you. Thank you, everyone. Good afternoon, dear doctors. It's my honor to be invited and give a speech at the AAPS 78th annual meetings. Actually, I want to tell that it's the first time I prepared the draft for the speech because I really want to show a special speech to all the doctors here. And the title of my speech today is Origin of SARS-CoV-2, Mandates and Communism. We are always in the front lines, doctors. I'm a medical doctor and an independent virologist with over 5 years of experience working in the WHO H5 reference lab at the University of Hong Kong School of Public Health. My research expertise include SARS-CoV-2, influenza, and vaccine development. One of the well-known identity of mine is that I am a whistleblower. From December 31st, 2019 to mid-January 2020, at the behest of the WHO reference laboratory, I conducted a confidential investigation of the outbreak in Wuhan. I used my personal connections inside China's research and hospital systems, as well as the Chinese CDC. The first-hand information I obtained, plus my expertise, allowed me to understand not only the severity and the transmission potential of the COVID-19 disease, but also the non-natural origin of its responsible pathogen. I also learned that the Chinese government was hiding information which was deeply worsening the situation. I then decided to expose the information, hoping the pressure from the international community, which could result from this exposure, may force the Chinese government to take responsible actions and prevent a global health crisis. On January 19, 2020, through Luda Media on YouTube in Chinese, as I show, I anonymously shared the information that I uncovered, including my own analysis on the origin of the virus. My messages was the following. The virus was created in Chinese military laboratories using bad coronavirus ZC45 and or ZXC21 as the template. Remember, not RATG13. I explained in my reports that is a fabricated one they submitted right the next day after my revelation. The second point is the Chinese government was actively covering up the truth about the disease with WHO. The third one, there was human-to-human transmission. At that time, both China and WHO denied that. And since we delivered the message out, China government has to admit it within 24 hours. That's the first time you know it in the world. The fourth one is there are no wild animal intermediate hosts, and the Huanan seafood market is not the origin of the virus. No matter the things you heard that we Chinese may eat, pangolin, bat, snake, raccoon dog, trust me, they are not intermediate host, and we don't eat bat. Yeah. I mean, my friends, all the doctors, all the people I know Chinese, when they see the photo that people eat bats, trust me, their reaction are as same as yours. The fifth is, if not controlled immediately, the virus may lead to a pandemic, and as a result, various mutants will emerging inevitably and quickly. By the way, Mr. Lude, the top influential Chinese anti-CCP blogger, is here. He's company with me here. We are both the Chinese fox hunting target. They call us foxes. All 5 messages have later been proven to be true. With its plans disrupted, CCP government reacted in a hurry. I fled to the United States in April 2020 and soon started alerting the non-Chinese speaking world about the true origin of SARS-CoV-2. Furthermore, since September 14, 2020, I and the co-authors of the Yin research have published 3 Yin reports, which proved scientifically the laboratory origin of SARS-CoV-2. These reports are rigorous and have been proven unassailable by attempts that failed in challenging them. These are the 3 Yin reports. You can find it on the nodal, and the views of the first Yin report about the lab origin has been over 1 million in the original website. And altogether, China government cannot do anything to refute it until now. In this report, I tell people what have been done in the virus, how they did that, why these scientists, the Chinese military, civil, and also their partners in America has done that, because these are all proved in their own publications. And also I mentioned that misinformation actually is organized by Chinese Communist Party behind. And also this virus is not only a virus, it is an unrestricted bioweapon which China planned for years. People always ask me why I decided to take the huge risk to speak out the truth of COVID-19. Let me share with you a story. In mid-January 2020, I called one of my friends, a neurologist in Wuhan. I asked about the situation in her hospital. Guess what? She was very nervous, almost cried, even dared not to tell me anything initially. I promised that it was a confidential conversation, and the information would be provided to WHO experts for reference. Then I heard that all the doctors there were helpless. The government forbade them to discuss the novel virus, while the number of the highly suspicious patients was growing like crazy. There was no protection for doctors, no treatment for the patients, and the CDC refused to give the results of diagnosis. Because at that time, the Chinese government insisted on only 47 cases in Wuhan. The doctors didn't know what to do and how to do it, and they couldn't even protect themselves and their families. They are vulnerable. I knew the Chinese Communist Party never cared about human lives. I knew the novel coronavirus in Wuhan is a gain-of-function product from Chinese military labs. However, when I saw the doctors were forced to expose themselves to the outbreak caused by the lab-enhanced virus, it deepened my understanding of the evilness of communism. The doctors, no matter my friends or strangers, are all cheated by CCP's cover-up and lies of the COVID-19 origin in the front lines of that ongoing crisis. I must find a way to help them. I keep revealing the origin of SARS-CoV-2 and CCP's non-traditional bioweapon, which I defined as unrestricted bioweapon program since then. The documents I obtained from the People's Liberation Army clearly demonstrated that Chinese Communist Party has spent over 20 years studying such strategies to achieve world domination by 2035 or earlier. Among the strategies, mandate is a typical tactic in communism ideology to carry out its totalitarian policies. Meanwhile, it is a pragmatic approach to division. To pro or anti-mandate, Americans are separated spontaneously. Let's take the COVID-19 vaccines as an example. The vaccines were originally designed and given emergency use authorization based on an wrong assumption that SARS-CoV-2 came from nature. The misinformation of nature origin and organized propaganda from the scientific community, which is infiltrated and manipulated by CCP, brainwashed people. While it was ignored that the virus was genetically engineered, with known and still unknown functions designed to target humans. It is a foundational flaw in the development of COVID-19 vaccines. It is proven that the COVID-19 vaccines cannot stop transmission. On the other hand, the lack of data concerning vaccine safety, as well as its efficacy against the ever-emerging variants, could lead to serious consequences. Instead, we know there are safe and effective protocols using cheap drugs like hydroxychloroquine and ivermectin in either prophylaxis or early treatments against COVID-19. Nevertheless, we are threatened to leave them alone. The doctors are obliged to accept and promote vaccines to patients in the front lines of the mandate. It is a tragedy for American freedom. I grew up in mainland China. I know how the Chinese Communist Party controls and harms Chinese people. Then I moved to Hong Kong for 8 years. I saw how awfully the Chinese Communist Party regime turned Hong Kong into hell in a short time. They won't stop until one day that America is in CCP's pockets, and Americans worship the Marxism and communism ideology. SARS-CoV-2 is one of their best tools to undermine the United States. The mandate is another tool. One key topic of the International Report of Biosafety and Biosecurity, as I show, published in 2020 by CCP, is to further manipulate the prevention and the control of emerging infectious disease using Marxism communism ideology combined with the technology innovation to accomplish CCP's world domination in the near future. They sit on their trophy in the COVID pandemic, looking forward to destroying the economy, social disorders, and the civilization of the United States. We are in the front lines against the vicious communism. Dear doctors, I hope my speech would provide you better insights of the real danger we are facing nowadays. It is neither a joke nor conspiracy. It is the facts. In the eyes of Chinese communism, the doctors are always in the front lines of being sacrificed. Please, for our safety, our families, and our next generation's future, let's work together to fight against the CCP. We don't have much time, but we can always be in the front lines. Thank you. Thank you. And I want to thank AAPS conference. I want to thank the Yen research team. It's an independent coronavirus research team, and we do everything for volunteer until now. And also Dr. Craig, Dr. Richard, Jeremy, and all the people who are helping me to get here and talk with all of you. And you can follow me on my Twitter, but please remember, there are hundreds of fake accounts established by the China's cyber army, and this is the only one with over 100K followers. Thank you.
Have a seat.
Let's direct a little bit for Dr. Malone. He's got a
Sure. Okay. Sure. Okay. All right. So we're going to have a quick question and answer. We're going to try and keep them brief, keep them short. Initial questions for Dr. Malone, because he has to catch a plane. We're going to go first. So any questions for Dr. Malone?
Yeah, I have a question. The first question, Novavax, is that a spike protein that's being developed, or is that like the other one that you talked about? And then I have a question for Dr. Cole. Lymph node biopsy, getting stains on a lymph node biopsy for spike protein. Thank you.
Novavax, as I recall, is spike expressed from insect cells formulated as a virus-like particle, and with a saponin-based adjuvant. It's currently stuck in advanced development consequent to problems with quality assurance and quality control concerning harmonization of all other manufacturing facilities. When it's going to get out of that, I don't know. It appears to have been put on a status where BARDA has withdrawn the funding for the product, and I don't know what they're going to have to do to recover from that. So I have no insight into what the timeline is going to be for Novavax.
Okay, another question for Dr. Malone due to time.
Sure. Robert, we use recombinant vaccines, which we like because they're totally clean, such as the Heptivax. RNA vaccines are a different animal, isn't that true?
Yeah, RNA vaccines and the advector vaccines are both gene therapy technology applied to the indication of vaccination.
Right, so we don't get them confused.
They are not subunit vaccine or purified protein or the products that are closest to RNA vaccines are the live attenuated vaccines such as yellow fever.
Okay, thank you. Next question for Dr. Malone, please.
Dr. Malone, I have a question, might overlap to the others, but it's been said vaccines get on the vascular endothelial cells and might have a connection to the impact of the type 2 pneumocytes in the lungs, and why lisinopril might dismantle the COVID from the type 2 pneumocytes. Do you have any thoughts in that regard? And Dr. Cole or the others, have you used lisinopril for the long-term COVID with regard to the type 2 pneumocytes?
So you're talking about the spike protein released from the RNA vaccines, or are you talking about the pathology of the virus?
No, the spike proteins attaching to the vascular endothelial cells downstream affecting the type 2 pneumocytes in the lungs.
So what you're basically asking is, are the cleaved products of the vaccines which circulate, we know from the Harvard and Brigham study, are they able to bind ACE2 and trigger the effects associated with ACE2? To my knowledge, that is an unknown because of the 2 amino acid mutation in the vaccine product. Personally, I find it shocking that it has not been investigated to the best of my knowledge, but I defer to my other colleagues, knowing that we've all been thoroughly fact-checked, and that the press finds it difficult to discriminate between the toxicity of the spike versus from the native virus versus that of the vaccine, which has the 2 amino acid mutation. Please.
I think that's a hard question because I think it's still a riddle. I don't think this is an endothelial disease. I think that's a misnomer. We'll see as time goes on, but I don't believe it's an endothelial disease. I really do think it's a disease of the pericytes actually creating fibrosis and creating injury within the whole vessel system. And actually, Dr. Malone explained it to me really well earlier because we were talking about it, how it kind of, the arborization process, and it kind of clamps down, and that may be part of the injury and stuff.
So Richard is speaking of one hypothesis for the pathophysiology, and I'm sure Dr. Cole has his own insights. Yeah. I can't imagine, get 3 pathologists in a room and it's like lawyers, you have 5 opinions. One theory is that these smooth muscle-like cells, which control tone largely in the capillary beds, and this whole field of pericyte biology is still really in its infancy, but there's a logic. An observation is that the erythrocytes actually have a larger diameter than most of the capillaries do natively, and that the erythrocytes are basically squeezed through the capillaries using peristaltic action from pericytes. So the fact that pericytes have ACE2 receptors and are probably reactive to binding by spike yields the hypothesis that what might be happening is kind of like a chronic agonist activity where it's causing these things to contract chronically and block blood flow, which in the microcapillary beds, which would lead to microcoagulation, which is the key feature of the disease. So that is one working hypothesis.
Thank you. And Dr. Cole, did you want to
say one more thing?
Oh,
about
okay,
it.
Dr.
I
Urso,
think
then Dr. Cole.
also, because of the lipid nanoparticles can get through the tight junctions and get to the pericytes. I think that's why you're seeing the vaccine injury, although, I don't know if that's true, but that's what I think is the leading cause.
And in the laboratory, yeah, we're putting a lot of different tissues together to stain, because we can stain for the pericyte versus the endothelial cell, and we can stain for spike protein, we can stain for the different subunits, we can stain for actual virus itself versus just the spike subunits. So, we have the studies underway, at least in my lab, and I've been collaborating with doctors in other countries as well. So the short answer is, we don't know. I think it's an excellent mechanistic explanation. In addition, you have to look at the CD147 receptor, which is ubiquitous in the body, and endothelial cells, platelets, red cells. That's why ivermectin works so well, is because ivermectin displaces viral subunit off of the CD147 receptor, causing disaggregation of clotting. So that's a study we will continue to do and look at.
All right, thank you, Dr. Cole. Is there another question for Dr. Malone?
Hi, Dr. Malone. I wondered, since you've had research with DoD and DARPA in the past, your thoughts on the current vaccine research utilizing self-amplifying RNA or that based on the VEEV backbone?
So actually, we were out front on that in the early 90s, and that was the research that led to the patent on mucosal vaccines with Peter Berglund, who had trained with Peter Lillystrom at the Karolinska and was a postdoc in our lab. So the problem, so the alpha virus vaccines, which is really what you're talking about, they have had a spotty track record in terms of toxicity, particularly neurotoxicity. Now I don't wish to cast shade on my good colleague from the Karolinska, who is, I think, on the member of a committee that's going to make an announcement on Monday, so just have some forbearance. But it has been Peter Lillestrom's leading position in developing that tech. And alpha viruses and alpha virus vectors for sure have a chronic history of neuropathology that has compromised their utility for gene therapy in the past, and I can't predict what may come in the future. Have I answered your question?
Kind of.
Okay, to some degree. Okay, next question, please. Brief, please.
Yes.
Dr.
Thank you.
Thomas.
Adjuvants. Adjuvants can be secret. Matrix M, used by Novavax, can have secret ingredients. The intelligence community hasn't weighed in on what they've put in and what they haven't put in. There's one company that doesn't use adjuvants but uses electroporation instead.
Yeah, I helped found that company. It's based on my technology.
How wouldn't that be better, where they couldn't put in these
You're
quote,
talking about
secrets?
Inovio, and there's actually another electroporation company also in San Diego. The pulsed electrical field work, I think, has fascinating indications for rapid response in small populations. So the DoD is a big fan of this for special ops command type applications. But for global deployment, not so much. I don't know if you've ever electroporated yourself or participated in a clinical trial using pulsed electrical fields for gene delivery and vaccination, but it causes muscle contractions, and that's a general way of putting it. The patient's satisfaction has been rather low with the technology, and there are ways to circumvent this, but the efficacy of the Inovio product was championed by Francis Collins during the Zika outbreak, and those vaccines turned out in practice to not be very effective. You'll recall also that Inovio tried very hard to, and actually met in the White House to advance their own product for this outbreak, and that has not been as successful. In contrast, there is a DNA vaccine, a traditional DNA vaccine by needle injection, that is currently authorized in India. So the DNA vaccines are moving forward, and they don't necessarily need to have the pulsed electrical field. I hope if you wish to discuss that further, you can look in my papers or others, but I had high hopes for pulsed electrical field based vaccines because they do get the polynucleotide and dendritic cells, but that doesn't seem to have panned out, and I don't think it's going to be very practical for mass deployment.
All right, thank you, Dr. Malone. If you need to catch your plane, we'll continue. So, but thank you kindly.
Yeah,
another
thank you,
hand for
Dr.
Dr. Malone. Okay, we'll continue with questions. We're not quite finished. Please, yep. Okay, so there's one question from someone watching at home, and then we'll return here to the studio audience. So this question is for Dr. Yan. Why are the COVID counts different in the United States versus China? Dr. Yan, why are the COVID counts different in the United States versus China on the basis of how deaths are measured?
Oh, that is a very good question because many doctors and also I think even some policymakers in America ask me that because Americans seems really believe that China can control COVID, but Americans fail to do that. Let me tell what happened in China. China doesn't want the other country feel that they cannot control COVID because propaganda is so important for the China government. They want to use this COVID-19 virus to destroy your faith to your country and try to turn your interest to the dictatorship China style. So let me tell you how they make the numbers so low in China.
Listen, guys, please, we need a little quiet in the back, please. Thank you.
As I told people that from the beginning, China government denies the cases. They don't allow patients to get diagnosed. They don't allow doctors to talk about highly suspicious cases. In the lockdown in Wuhan, it's not like the lockdown you see in America. They just lock people in their residence buildings and let them die even there. There are so many people, they suffered, they died, and with even not a number in the Chinese record. And also in China, at that time, I was in Hong Kong, and I almost the first one in the world to conduct the autopsy about COVID-19 in early February last year. But that project, when we finished all the preparation, was rejected by the China government because China government doesn't want anyone to say they have the COVID patient death case at that time even. I mean, the autopsy will tell more secrets, right? So China, basically, they claim that even if they give us the dead body for examination, we cannot publish the paper that this is a COVID patient. How they have done, the doctor for that patient told me that they put the doctor in the ICU, gave the ventilator, and until the day the negative result come out from the swab, and government will tell them, okay, it's done, this is not COVID patient, and this guy is not the number. So then these people die because he's almost dying. When it becomes negative, he died, and he is recorded as someone died of the heart failure or the other problems. Yeah. So that's why the death toll in China is also very low. And also for the PCR result you are using in America, although not 100% accuracy, but in China it was 20 to 30% accurate. So that means for twice examination, you still cannot see the real result. Most people are false negative. And also China government will tell you that we don't have cases. Now recently, one thing I can tell you is what China government do is they tell people in China 0 cases. However, they hold the people from foreign countries, even the Chinese citizen, in a special place for first isolation and then do record. Then after that, for example, this guy went to Beijing. He stayed there for 80 days, and At the 80 days, suddenly Beijing government feel, okay, there is outbreak in Beijing, we cannot stop. And then they will trace back to this poor guy and said, hey, this guy come back from America 80 days ago. And yeah, he was asymptomatic at that time. Now he is the origin of our outbreak this time. It become imported cases, and China is not responsible for that.
Dr. Yan, can I kind of repeat that back quickly? Yeah. So there were two issues. One is they used a different amount of cycles with the PCR test, and I think Dr. Cole can speak with that. He spoke on my radio show eloquently to that. The other thing is the way they count cases here and the way they count them in China. From my understanding of what you said, is in the United States, once you're positive, you're always positive no matter what you die of. So if you are positive within a month of dying, no matter if you die of a heart attack, a stroke, a cancer or something, you're considered a COVID death. So that raises the rate in the United States. In China, they did exactly the opposite, that if somebody was positive when they were admitted and went on and suffered for 2 weeks, at that point they would get a negative test and then say, oh, it's a negative test, it wasn't a COVID death, it was something else. So that's why the numbers in the United States were so high, the numbers abroad in China were
Yes.
relatively low. Is that correct?
Yes.
Okay, Dr. Cole.
And to that point here, if you look at what the CDC said last year, pure COVID deaths were only about 6%. And then if you look at, again, cycling the PCR, how many false positives, how many false negatives, running a PCR above where you could even culture virus, which ends at about cycle threshold 30-ish, you don't have meaningful virus. And it's more meaningful the lower your threshold is. Yeah, data here in the U.S. is very broken, very retrospective. Just because it came to mind, one thing that we're not looking at, if you think of these individuals who die, where a perfect, healthy young man, young woman dies of COVID, there's a great study that came out just a couple weeks ago, and it looks at anti-interferon 1 antibodies. The virus hijacks your interferon. And in order to have a good antiviral response, you need interferon to tick upward. Ivermectin is one drug that does that. The interesting thing is these healthy patients that do very poorly, it's a good percentage of the population. In the elderly, it can be up to 1/5 of the population that has a self-antibody against their own interferon. And the younger patients is like 6 to 10%. I just wanted to go onto that tangent because I just haven't heard that shared very much. And it was a 35,000 patient study. It was a big study. And so it's interesting how we panic over the deaths here and attribute everything's COVID, and we don't think about the comorbidities, that 80% of America is insulin resistant and metabolically unwell, and all our underlying conditions. And so it's fascinating, here we cover up vaccine deaths, and there they cover up COVID deaths. It's fascinating, the dichotomy.
Sure. Is there something you wanted to add,
Yeah,
Dr. Yan, quickly?
One thing
before we
very
move on?
important is in China, they use hydroxychloroquine for treatment. And last February, the Chinese military scientists filed the hydroxychloroquine as a new patent for the new purpose to treat COVID-19 in China. And that got the top awarded by Chairman Xi at the end of last year. So that's also one reason why we have less death toll.
Thank you.
Early treatment saves lives.
Yes,
How
it's
about
true.
that?
This is true. All right, next question, please.
Yeah, Dr. Richard Dalinar from Phoenix, Arizona. What a spectacular panel you've put together. AAPS definitely should be congratulated. My question, though, is for Dr. Yan. I have two questions. First question is, how did that virus get out of that lab? Was it due to poor technique, or was it intentional?
That's a great question that's on everybody's lips.
I have a second question for follow-up.
Okay, it is intentional. Get out of the lab, but it was not intentionally launched the bio attacks in Wuhan. So the thing is, according to all the evidence, and I analyze and I provide to the certain department in America already. So this virus, when they design it, definitely is to attack people, and they try to make it optimized. And according to Chinese Communist Party's document, which made in by the People's Liberation Army scientists, they mentioned that the ideal, this non-traditional, unrestricted bioweapon, would be looks like nature occurring. That is the key point. And once it is revealed, the thing is they need to use misinformation and denial. That's clearly in their document. And then also, At that time, they mentioned that they need to optimize it as everlasting virus, pathogen, I mean. So the best way is to test it in a totally identical environment as enemy's area. That means something like neighborhood cannot be in the lab. And also, at that time, the Wuhan lab, they got the virus. They are not the only lab made it, but they got it at that time, and they try to make it more optimized. And then they try to do it in the neighborhood in Wuhan. And at that time, because they underestimate the high transmissibility, they don't have the right animal model. I'm the one published that on Nature last May, but before that, no one sees a real animal model to test the high transmissibility, and that is not the parameter they designed. That is something combined after their gain function enhancement. That's why when they do that, and it is out of control. And at that time, China claimed it's under the control because they underestimated. That's how it happened.
All right, thank you, Dr. Yan. Next
My-
question,
my-
Dr. Dolinar, briefly.
My second question. So you've been in the United States for at least a year, if I understand correctly.
Over 1.5 years almost.
Okay. And I understand you probably have spoken with government officials at our highest levels of government. If that is so, why is it during the last few months when they were, quote, looking for the source of this virus, et cetera, nobody came forward and said that this is definitely coming out of the Wuhan lab? They seem to back off from that. Do you know why?
Well, there are two main reasons. One thing is this is misinformation campaign. It is part of this unrestricted bioweapon. Without it, people would already say it's from the lab. But with it, they dare not to say it. That's what Peter Daszak's work, and that's what Dr. Fauci's work, Lancet, Nature, Science, they all do together. And because we go through the media review it from January to April, even before I leave Hong Kong, China government has to hurry, in a hurry to push out all these things out. That's why now we can see the evidence and the mistakes they left. But still it affects because those scientists control the whole group. Especially in my lab, why I can see so many things, because my lab, we have the top coronavirus experts in the world. The guys, Malik Paris, behind Tedros, and behind the Lancet, behind all this, they control the power in this field. So this is deeply weakening the capacity of American and other government. And also the other part is they do know that in intelligence department, I can see the people I contacted. I mean, I cannot say where they're from. I even don't know some of their real identity, but whoever come to me, I just tell them. And they actually not feel surprised about this is special bioweapon. But the thing is, China spent years, more than 2 decades, spend their time infiltrate you, manipulate you, and also they use the weakness of the laws. They use the weakness of the Bioweapon Convention and the bioterrorism. There's a lot of things I can explain later for details, but the thing is they take advantage of that. That's why even the policymakers say this. They really don't know how to do that. So yesterday, the Senator Foreign Affairs Committee has put the bill to talk about, make the bill to stop this kind of threats from like China, this kind of country. That is a very good beginning, and I can say we are behind that to provide them evidence and the suggestions. They have to first fix it up and then hold China accountable.
Thank you, Dr.
Thank you so
Yan. And
much.
again, to recap, per Dr. Yan's information, which she's been publishing, it did come out of a lab. It was a volitional act. It's been 20 years in the making. It was specifically manipulated to do what it does, to attach to human epithelium and to catch. It's just they didn't know it would catch so far, so fast. Okay, right. It's a multi-system disease that it begins with the respiratory epithelium.
Yeah, it's not fair to just leave out the fact that there's many international people that are involved with this, though. I mean, it's not a Chinese entity all to itself. I mean,
Please.
clearly.
Dr. Urso?
Well, I'm not an expert on it. Peter Breggin has looked at this in detail, and if you guys know Peter Breggin, he's wrote a book on this, and you all should read it if you want to know about the origin of this virus, besides what happened in China, and we've used them as a scapegoat. but it's a team approach. The U.S. has been involved. People in Europe have been involved. People in North Africa have been involved. A lot of people have been involved in setting up this virus. I'm sure a lot of you have seen David Martin's work and all the patents that have been done. So, you know, it's not fair to just say it came out of one place. There's a scapegoat here. It's just to create division. And I think in general, if you hear the tone of everything, what you're seeing right now is we're still talking and fighting about things instead of talking about solutions. So we're talking about what happened, and we're looking back at ourselves and trying to figure it out. And instead, as doctors, we're trying to be detectives about what happened in the past. And in general, you know, we're not getting the message out on how to treat patients early, you know, how to create strategies for prevention. And instead, we get stuck on these issues. And I think to say it's one country that did it, it's not fair. So it's an international alliance that created this whole virus, that created the whole international attack on us. It's happened where we have people that are getting attacked by overseas journalists, people that are out for her when she's got to be careful everywhere she goes. So it's an international attack. It's not one-sided.
Yeah, I think that's definitely a fair statement, and unfortunately, there's a lot of involvement with our government and officials and politicians, because many of us have tried to be creative throughout the entire time frame, trying these new therapies, only to be stifled or limited by governors or federal government entities or faceless bureaucrats, and that's difficult. And as Dr. McCullough will probably mention tomorrow, his strategy is to keep on moving. You know, so they kill hydroxychloroquine, we move to ivermectin. They kill ivermectin, we move to macrobid.
I've got a comment on that. I'm sorry.
Sure.
I say that all the time. I say, you take away hydroxychloroquine, fine, I'll find something else. Take away ivermectin, I'll find something else. It's not that hard. It's not that hard. There's a lot of drugs that work.
And
Thank
it's
you.
not
Dr. Cole?
Yeah, it's not per se about a virus anymore. It's about an unwillingness to allow early treatment. We have a pandemic of under early treatment, and we have a malicious, I don't want to say malicious, but appears malicious to undertreat in the hospital setting as well for a disease that's clotting and inflammatory, and we have countless drugs we've used for decades upon decades upon decades that stop clotting and inflammation. And why we're not pursuing that and only giving money to a couple of drugs that the hospital gets big money for using is insane.
Yeah, and to go forward on that, none of these drugs are off-label. There's nothing off-label about using stuff for inflammation. I mean, all the drugs we mentioned today have anti-inflammatory properties. Some of them are approved for inflammation. And we're same thing with the antithrombotics. They're all approved. So we're on label with most of the things we're doing. We just went to try to go to nitazoxanide about a month ago, and the price for that, if you guys don't know, went up from about $100 a treatment to about $3,000 in a week, I guess, because there's only one manufacturer. So if that doesn't work, we'll go on to the next thing and the next thing and the next thing. There's a lot of those wonder drugs we talked about, and we'll have solutions.
Well, thank you, Dr. Urso. Next question.
I know this isn't a topic that you guys talked about, but I do want to backtrack to it because people actually ask me, and I want to try to give them as honest an answer as I can, and it does have to do with the vaccine. So I live in a community of a lot of older people, and they ask me about the vaccine, and they ask me about doing a third booster and whatnot. And the question that I even have for myself, as I understand it, that the Delta variant has to do with a lysine substitution, and that the vaccines, and I'm not a vaccinologist, I'm a psychiatrist, but that it was a very, very specific vaccine. So that if you had, number one, you had failure, and so if you had failure, why would you booster with something that's already failed? And then if the booster was specific, and if it was specific and we're having breakthrough on something that's failed, would the booster work? And number three, then I'm also looking at the Israeli data. It looks like the Israeli data is failing anyway. So how can I recommend, or should I recommend, and I'm a psychiatrist, I'm not going to, but I'm still a smart thinking woman, really what is the answer on for this elderly population? Who is the people that, are the people that are the vaccine recommendation? That's the number one.
Can I try and summarize that?
Yeah,
you're asking about who should be vaccinated, who should potentially consider a booster, and is there any rationale at all to doing the same thing 3 times and expecting a different result?
Right, that's number one.
Okay.
And then number two would be a comment to you, which I've wondered about since I first seen you on the scene, which is you must be terrified. I can't imagine how you could come here with the information that you have and not be just terrified every single day of your life. I just can't imagine that.
You know, just
Okay,
to
so we've
repeat
got
that,
two quick questions on the
Yeah,
floor.
hold on a second.
Okay, sure.
She is terrified, I'm sure, you know, but at the end of the day, everyone in this room should be terrified, okay? Everyone.
And the whole world should be is another
You know,
thing.
it's just like what happened in the past. Today they came for her, tomorrow they come for her,
Right.
then for him, and then for me. That's how it's going to go. So
Right.
if you're not terrified, you should be terrified. And she's brave, and that thankfully, she's terrified, just like we all are.
Right, if you're not terrified, you're not paying attention.
And then
Okay.
the other thing is AAPS, I would like to speak with you. We have a statement. I would like to spend like 2 minutes with you and just have you look at our statement.
Yeah, afterwards, that
Yeah,
would be great.
afterwards,
Sure.
obviously.
Yeah. Dr. Cole?
Yeah, I mean, fear has been, what did Hermann Goering say in the Nuremberg trials? How did 9% of Germans overtake 93%? Easy, it was fear.
Yep.
And, you know, let's flip it on them. Fear, freedom, E, early treatment,
Love it.
A, America, R, we have our rights. That's what fear should be. You know, flip fear on them. We're a free people. Yeah, terrified. I got a board complaint from the Idaho Medical Association today because
We've got a great lawyer here.
Good, yeah, because they complained that I treated patients with ivermectin, and I'm a pathologist. And never mind that I worked ER for 4 years back in the day.
Right.
And I'm not a doctor anymore, they say, whatever. But anyway, back to the vaccines. This is a tough question. The vaccine was formulated for the Wuhan spike that's not even in circulation anymore. It left circulation January, February. If you look at the relative risk versus the absolute risk reduction, the statistical game they played with these, it's really, you know, Moderna was like 1.1%, your absolute risk reduction, and Pfizer was 0.7. Now with Delta, they're far, far less than that. I think we did a calculation that's like, yeah, they'll fact check me, I don't know, but fractions of that. So boosting with the wrong protein against the wrong virus that's in circulation doesn't make any immunologic sense. The second point of that is what Dr. Malone didn't talk about. In immunology, it's called high zone tolerance. You get a shot, you get a shot, you get a shot, get a shot. Guess what I do for a kid with peanut allergies back in the day? Well, I give him a little bit of an antigen, more of an antigen, more of an antigen, until he can tolerate that antigen. So you get chronically exposed.
Sounds like being an allergist.
Yeah. So you get exposed to an allergen constantly, and all of a sudden your body says, I quit. I'm not going to recognize that antigen anymore. So if we play this whack-a-mole booster game, eventually your body's going to say, well, I'm just not going to react anymore, be it a T cell response, be it a B cell response. Doing this makes no immunologic sense over time.
All right, thank you, Dr. Cole. We also have another question from home based on what you were talking about. You had said something about absolute risk reduction versus relative risk reduction, and if you don't mind spending a moment on that, because a lot of times, you know, when people say, oh, it's 95% effective, but that's smoke.
You take 30,000 people or however many it was in two groups, and you say, okay, 16 people got COVID in the vaccine group, and 100 and whatever got it in the other, and then you divide and you go, okay, there's your difference. Well, you're ignoring the 30,000 people, so really it ends up being a tiny fraction of that you're protecting, and you know, like.
So relative risk looks larger than absolute risk when you consider the entire population.
The numbers for Pfizer, the relative risk was 95, Moderna 94, AstraZeneca was 67. The absolute risk was for Pfizer was 0.7, 0.8 for Moderna, and 1.0 for AstraZeneca. Those are the absolute risk reductions in those early trials. So the trials actually did not show much absolute risk reduction.
And
Thank you. That's exactly what I wanted to get out in black and white. Thank you.
If you look at their, quote, efficacy now, Mayo did a study back in June. It was 42% for Pfizer, and then Israel was 39%. But that was before Delta really took off. So now we know those are far lower than that, and that was relative, not absolute. So that's why my comment is it's far lower now.
Thank you.
And if you looked at the data from the Pfizer trial, if there's a great thing in the British Medical Journal has a great analysis of that. Yeah, it was Peter Doshie's, if you want to look at that. Basically, he calculated the real relative risk was about 19 to 29%. One last thing about vaccination, which maybe I should let Dr. Cole say this, but vitamin D, I found this out from hepatitis B vaccine. So everybody here had their hepatitis B vaccine. Has anybody lost their antibodies from not having it, any hepatitis B? you're likely vitamin D deficient. So if you're vitamin D deficient, you're likely going to lose your antibody production, your immunity production. So, do you want to comment a little more on that?
100% true. In order
Then we need to move along.
stimulate your CD4 cells to talk to your plasma cells and your T cells, your immune memory is dependent upon your vitamin D level. And so this is insanity. We're vaccinated. Why did it take 2 shots to get an immune response? And why do we have certain shots that take 2 shots? Because we have a population that's 70% vitamin D deficient. We wouldn't have to have such high dose vaccines if we had normal D levels. So people lose their immune response due to deficiencies that are easily correctable.
Okay, Dr. Yan?
Yeah, I will comment something about the vaccine. I mean, when people come to me ask whether to get the vaccine, I will tell them I don't get this vaccine because I want no harm first, and also I have drugs to protect me. But the thing is, I won't tell them to do what or not to do what. I will tell them pros and cons. I will tell them it's like drinking the alcohol or smoking. You know this is not safe enough. We know there are potential things, potential risk happen, and we know it can have some consequences for some people. But if you feel that can release your, remove your panic, then you do that. You take the responsibility for that, like you prefer smoking or prefer drinking. And also for the vaccine, actually, I mean, there were potential antibody dependent enhancement, ADE. And also for the SARS-1 virus, they have found that allergy and other side effects, which they cannot overcome. That's why their vaccine failed. But now they push it to SARS-2, which is a bigger version of the enhanced version of SARS-1. So we can imagine what's the harmful or what's the potential risk inside. And also, as we mentioned that, doctors mentioned about the variant. So they made it when there was Wuhan strain and also Alpha strain. Now it's already Delta. Basically, people get it now. Even booster, it's always eating the expired food. It makes you full, but it also can make you diarrhea. But it's not a good quality food, right? Expired already.
Right. Okay.
So yeah, and also I want to thank people's care about my safety. It's tough, but I always tell people that I keep fighting until the last moment, either me or the Chinese Communist Party. Thank you.
Thank you. Thank you. All right, next question, please.
Hi, I'm Dr. Kerrigan. I'm a internist here in Pittsburgh. I've been treating successfully COVID with ivermectin or hydroxychloroquine up until about a week, 10 days ago. Now I can't get any pharmacies to fill it. And you touched on this a little while ago. In light of that, what combination of the medications that you put on the screen would you recommend to use instead of that?
And then can I say one thing? That's assuming that you can't get it from a local mom and pop pharmacy, the few that are left, or a supermarket pharmacy that might be more flexible than, say, CVS or Walgreens, which has algorithms that block them.
Yeah, I've talked to the local, and they're
and there's also like Blueberry Pharmacy and things like that are mail order that can get them to you the next day. Don't fall in love with Amazon for that. I had a patient that said, oh, I'll just get it from Amazon. The next day, they sent it back to me, and they said, oh, we need diagnosis codes for these things. And if any of them are COVID, we don't fill for COVID. So they not only delayed care at least a day, but then refused to do it. So,
that's what
but there are some that will. Go ahead, Dr.
Yeah,
Urso.
so we've had this even in the hospitals, same thing. Many of the hospitals won't use hydroxychloroquine or ivermectin, as you know. Been able to get hospitals to do phenofibrate. I think that's a great one right up front because you're going to get up to, in the tissue culture cells with viral kidney cells, up to 70% reduction in viral load. Plus, you're going to get all the downstream anti-inflammatory effects. So that would be one. And then you'd put in azithromycin, definitely a macrolide. Started right off the bat. It has antiviral effects and has all the immunomodulatory effects that you'd want as you go downstream. Ciproheptadine, serotonin blocker, H1 blocker. Pepsid, high dose, maybe 40 Q12, and anybody over 200 pounds, 20 Q12 I do under usually. And then I'll leave a couple out. Yeah, aspirin, of course. But even so, sometimes, check the D-dimers if you worry about that. If you're worried about that, basically that's something else, that you can do is maybe Lovenox. I've gotten Lovenox, and done that at home. And so I would go with phenofibrate, azithromycin, ciproheptadine, famotidine, Singulair, go maybe 20 a day. Dutasteride works early on the viral load, blocking the TMPRSS2 serine protease. It needs that in order to sort of sit down on the virus. So that's another one that'll make a huge difference. And then vitamin D, load with vitamin D. If you haven't seen the study, basically they did loads, 40 to 50,000 of vitamin D for like 4 or 5 days, and it massively improves the downstream anti-inflammatory effects.
And
it also
no known
has
toxicity
it also
issue.
has any
Okay.
there's no toxicity.
Just
and
downstream.
no toxicity. And there's also downstream effects for thrombosis for vitamin D. So, and then also hit them hard with steroids, at least 1 milligram per kilogram as you get them into day 8. I usually start them early. I don't wait actually anymore. I was waiting and I don't wait anymore. I usually start them 20 to 40 right off the bat when they get symptoms, because basically by the time I actually get them, it's usually day 3, 4, or 5. So I'm not waiting anymore on that. I found that makes a big difference. If they start at day 8, 9, and 10, that's where you're really going to have a problem. And so I learned from a guy in South Africa, Dr. Shetty. He pushed the dose up to 300 milligrams. I've gone as high as 160 for like 2 or 3 days, and it knocked them back down. And that was a huge dose. And that's in people where a couple of them we couldn't get the hydroxychloroquine or the ivermectin, and we got some inflammatory reaction around day 8 or 9 or 10. And as I said, that was been a huge thing. Basically, that regimen I just gave you. The other thing is make sure, because you're using steroids, to basically do keep them no carb, tell them no sugar, no fruit, no Pedialyte, no Gatorade, because they don't know what that means. No carb. That doesn't mean anything to most people. Literally, I had a guy getting anion gap by not. I told him no carb, but he didn't know what that meant. He was drinking Gatorade like crazy. And he went to 547 sugar and anion gap, and I was like, oh my God, he's going to die.
Can I ask a quick question?
Sure.
What you had mentioned, steroids. So a lot of people use convenient stuff like
Dex-
a Medrol dose pack, which is probably
work.
too little, too short.
Yeah.
So how much is the range that you use, and how many days do you use
Milligram
it for?
per, I use it for at least 10 days.
Okay.
So milligram per kilogram. When I get to day 8, 9, 10, I go like a milligram per kilogram at least. So, and then if they don't respond, I go out and double it now. I did, it's all in the last, since Delta, it's been August. I've treated 503 patients since August 1st.
Wow.
So it's been really busy in Texas.
As
Dr. Cole?
far as steroids go, methylprednisolone actually has action against turning off certain cytokine genes. Prednisone would be my second. The third one is dexamethasone, which is fibromuscular steroid. So why the hospitals are using the weakest of the 3 that they could be using is nonsense.
And Dr. Urso, I figured you'd want to chime in on that.
Yeah, I
that
wanna
it's
chime
good
in on-
early, but not late.
I did stuff in traumatic optic neuropathy. I did some studies where actually methylprednisolone did have some cytokine antioxidant effects, actually. And so that was done 20-something years ago. Has a bigger effect than Dex did. But I like Dex a lot, too.
But you, Dr. Urso, I believe you had mentioned something about dexamethasone being used earlier, and like later in the hospital, there's some issues. Is that correct? Maybe I'm mistaken.
So I think the biggest thing is not using the high enough dose. I think that's it. And then watching the sugars is a big deal. And that's, I don't know if that answered the question, but usually,
Sure.
like if I do Dex, I usually do somewhere between 10 and 20 Q12 if I was in the hospital.
And if you can't get hydroxy, try Nigella sativa black cumin seeds. They have quinolones and quinines in them. And there's some studies out of Pakistan and India showing dosing on just your, grandma used to say in India, if you have a cold, take your black cumin seed. And you take your black cumin seed, and it has a spike bending effect, and so it inhibits binding. So there, artemisinin, Nigella sativa, there are a lot of natural products that will inhibit binding.
So the black cumin seed is for you, all you guys here know is chia pet. Okay, that's chia pet. That's black cumin seed.
You can get it at Whole Foods. You can get it at your natural grocery store, but quinolones and quinines, and at the right dose, you can inhibit spike binding to ACE2.
What's the dose again? I'm sorry.
I think it's like half a teaspoon. It's like 80.
Okay,
80
let me
milligrams.
just mention a couple of things, if that's okay, just housekeeping stuff. We're going to take 1 or 2 more questions, and then that's going to be it. However, if the guests will be available to come back for Dr. McCullough's panel tomorrow morning, we'd be more than happy to entertain some more questions. Okay, go ahead, Dr. Ammerling.
Craig, if I might just give a quick example to compare relative with absolute risk reduction. Let's say you have 2 groups of patients. You give one group a placebo, you give the other group a statin. At the end of your trial period, 3% of the treated patients had a heart attack, 4% of the placebo group had a heart attack. That is a 1% absolute risk reduction. Yet if you divide the 1 by the 3, it's a 33% relative risk reduction.
Thank you for
And
making that point.
And that is how they sold Lipitor to the world.
Yeah, thank you for making that point. Absolute versus relative risk reduction.
Dr. Li-Meng, thank you so much for your courageous stand. Let me
you.
just ask you 2 quick things. Was it not part of the strategy to push lockdowns? Because it wasn't enough to just get the virus out there. The strategy of lockdowns was aggressively pushed by China through the WHO, through friends in media and government around the world. And that is what really wreaked havoc on the Western economy, much more so than the virus.
Actually, lockdown is just one of the methods China tried to make to make America and other countries have economic problem, and later they will also the things they help it to do it is like the other first mandate make people divide, and also no matter mask, lockdown or vaccine, and also they use propaganda through all the social media and mainstream media to tell you your country failed to control these things and create panic and to make people lose the faith to the country. But the thing happened in Wuhan, that lockdown actually is because they really out of control, and also because of my revelation 4 days before, China government realized that some inside really come out and they are nervous, so they know they have to control it. That's why Wuhan puts the lockdown, and as I told you that it's different as the lockdown happened in the other countries. And also China's, there are lockdown in China, don't compare it with your American's lockdown. First, in China, they do very inhuman lockdown. They never care about human life. The other thing is, after lockdown, they just change the number of economic increase. So you see the GDP in China still keep increasing. But you know, lockdown always makes the economic problem, and China want it happen in America. Then they can narrow down the gaps between America and China to finally compete with you and then get the world domination. That is in their documents.
Okay,
So
thank you.
Next question. China funds American universities and university professors. My question is, Charles Lieber was professor of chemistry at Harvard, was arrested in, I think, March of 2020?
Yes.
For failing to report $50,000 a month income that he was getting from China for work with the Wuhan lab. What was he up to? Do you have any idea?
Actually, I want to say that is just one of the example from thousands maybe, this kind of cases. It's kind of open secret for Chinese, especially Chinese scientists, but I'm surprised that in America, people still feel it's something surprise. Okay, let me tell you that Wuhan actually, not only Wuhan Institute, also other institutes in China, most of them are military-civil fusion. That means no matter what time the government and the military want to control them and want to use them, they will be obedient because that's Chinese Communism Party. And also the scientists who are bring to China, actually they not only get the payment, as you know, that is very little payment for China government to bribe such scientists for the exchange of the advanced knowledge and network in America. That is just some visible part you can see. China helped the scientists get grants all over the world. If you check the Peter Daszak's email, he clearly mentioned China CDC head George Fugao and my ex-supervisor Malik Paris in Hong Kong will help him to become the membership in the NAS, National Academy of Science. And also, if you check the spokeswoman, Maria, I forgot the name, sit next to Dr. Tedros always. She is the lady sit next to Dr. Tedros in WHO to tell the COVID briefing. That lady gets a lot of help from our lab and also from China to be promoted with the publications to sit on that position. So these are reputations and also this kind of academic such corruption. So China can give them all this. China give title to those people. Dr. Robert Gallo in Maryland University, the HIV titan, he attacked me on end of September last year after I published the first report. he used very stupid words. I'm sorry to tell that, but
No,
it's
that's
wrong,
okay.
it's stupid.
Please be honest.
Yeah, to reveal that, and which I refuted in my third report. You can read it. But what happened? Just 3 months later, China gave him a top award in China. Basically, it means he can get free organ transplantation in China. So all these
Wow.
things, right?
Yeah,
it's open
just
secret
wow.
in China.
Yeah.
They have even hung out that kind of organs for Muslim groups. You know, so that's all the things why China can bring so many people to China, because attraction is really big enough, and that is country. So other people feel collaborate with China, it's safe. That's
right.
how it
Okay.
happened.
Thank you. Thank you, Dr. Amerling. Dr. Yaros, your last question, please.
Okay. The rollout of this bioweapon was mostly in areas where there was high 5G usage. There's a lot of speculation. Dr. Mert speculated that 5G is being used or will be used or is being developed to be used as a augmentation of the effects of this virus, or even the vaccines, maybe because people are talking about metallic things, graphene or other things being injected into it, which can turn your body into an antenna or control your thoughts or anything. Was there anything that you're aware of about the use of 5G as a adjuvant to the effects of this virus that the Chinese were planning?
That's a great question for the panel. So we're looking at special adjuvant use, things like graphene, things like that can be electromagnetic, and other things that we may not even know about that are there. Supposedly the adjuvants are the special sauce of an injection like that. So what are the possibilities for electromagnetic signals and a possible connection to 5G? Because we hear a lot about this, and it's easy to dismiss. However, is there a possibility even?
I'm going to answer first because I've done some production for a biologic. Stuff gets in there. When you ramp up out of nowhere, stuff gets in there. The beakers, the little piece of glass, little piece of rubber, that's likely what it is. I don't think there's any 5G in there. I think it's just production issues.
Can I go back to my Air Force Academy engineering days?
Why, yes, you can.
I would like to. So the strength of a wave is logarithmically inverse to the distance you are from a signal. So the amount of power coming out of, be it 3G, 4G, 5G, yeah, it'll fry a honeybee in front of it, but if you're 100 feet away from it, it's about as strong as a radio signal or a few gamma rays we're getting through, the building right now from the sun. So I don't worry about those things, what is or isn't in the vaccine. Sure, they have their secret sauce and secret ingredients. All I know is there's damage done by the vaccines, whether it's the RNA, whether it's, some of the adjuvants, I don't know. We're studying them. The magnets, I can't explain. Some people do, some people don't. Some people hypothesize some of the batches and lots are different than other batches and lots. I don't know. I think at the end of the day, if you look at the number of injuries, and granted, most people are fine, but the statistics of how many people aren't is the important thing. To have a medical therapy biologic product on the market with, 5 times the death of 9/11 is really kind of crazy that it's still out there. When we know that early treatment saved lives, and if we could emphasize the message that, other countries, what South Korea do, hydroxychloroquine, what's China doing, hydroxychloroquine, what are other countries doing early treatment, what India do in Uttar Pradesh where they have 240 million people and only 20 cases a day, blister packs for their whole population of ivermectin and supplements. So the rest of the world seems to know how to treat this, and we don't have the political will here to do it for no good reason. I have no idea why.
Right. Right.
So,
sight gag for those of you at home.
Yeah, that's a little hand motion there. Yeah, so, the question is, yes, there's something in the vaccines. Yes, there's RNA in the vaccines and DNA in the J&J, but at the end of the day, are they damaging? Yes, they are. So it's a risk-benefit analysis like she said earlier. If some people puts their mind at ease, and like Dr. Amerling said, your absolute risk reduction.
Thank you. Dr. Urso and then Dr. Yan to give the final message, please.
So My thought is, you know, these proteins, I mean, the things you can make with messenger RNA, there's no reason to add extra stuff in there. You could design it up to make a lot of different products. If I wanted to design something up, I wouldn't do it with a piece of metal or graphene or anything like that. You could just design up the messenger RNA to do whatever it is you want it to do. So it doesn't make that much sense, and production is a big deal. The drug that I ended up having get to market had huge production issues. It took like 10 years to get over the production issues. When you ramp up over a short period of time with this, something that's never been made before, it's probably a production issue. That's what you're seeing in those vials. I would say stop thinking about graphene and all these other things. It's stuff in the vial, you know, it's production issue stuff. That's what I think, and I'm pretty confident that's true. You can do them, if you really want to be a conspiracy theorist, you can make the messenger RNA do a lot of bad things. You can turn tumor genes on and off. There's a lot of things you can do to kill people just with messenger RNA technology. You don't need to put extra special sauce in there.
Okay, and Dr. Yan, if you don't mind, two quick things. One is if you could address that issue with the vaccine and the adjuvants and what your thoughts are on that design. The other is any final message to all the physicians and people watching?
Okay, thank you. So first thing is about the vaccine and adjuvant. I have my influenza universal vaccine patent impending, and I worked for that for 5 years. So I can tell people that, yeah, no matter what vaccine, even it's a safe vaccine, you know, when we talk about the vector, when we talk about technology, when we talk about adjuvant, there are always potential risks, and it's up to the individual response usually. But here, when we talk about the COVID vaccine, the most important thing is, I want to tell people it's not only about technology. No matter it is that kind of virus vectors or the mRNA technology, the most important thing is, remember, as I told you, the military lab has a bad coronavirus ZC45. And that one's S protein, compared to the SARS-CoV-2, the S protein, the Wuhan strain, it has almost a 40% difference. And this 40% difference, I have analyzed that, and many of them, now for me, I cannot see the meaning. And from the amino acids, they change. Seems not functional change. They seems just like to cover up their change. You know, it's like you copy other people's homework. You want change a little bit words, make it look like your work. But there are also very important functional part. So we identify that receptor binding domain is a really change which copies us once. Expansion is a core part. And also we identify the furin cleavage site. And also we identify that, so there are suspicion, not only us, that database, some data from the clinical samples from China, America, and Japan, they show that they see the antibody-dependent enhancement happen in the people's serum. And also these are all happened in the S protein. And China spent over 2 decades, including my lab as a leading lab, to discover the dengue virus, Western Nile, Ebola, Zika, high pathogen avian flu, all these. And these are all RNA virus. And they learned the knowledge about that. And they like playing Lego. So until now, still there are some parts. My team identified this is functional, should be functional changed. And we still don't know because now we have no lab to do such experiment to exam. Somehow, we see those parts are suspiciously connected to the neurotoxicity, that kind of function. And because there are also other data confirm there are some antibody that can be related to that shown happened in human. So these are the things I cannot tell a lot here, but what I want to tell people is there are a lot of unknown functions, and I have identified the network in the Chinese military medical academy. There are labs working on peptides, working on dengue, all this brain damage, all these things, and working on how to change this virus and shift it away from different virus. And you can imagine this all happened in spike protein because there are still a lot of things we don't know how to explain. And China keeps the envelope protein 100% identical between the bad coronavirus ZC45 and the SARS-CoV-2 virus. That is evidence that China want to maintain the function of the E protein after their modification for the virus. And the E protein can maintain the neurotoxicity. That was found by our lab before. It has some of this function. So all this tell you that when you get S protein, you have to think about what else inside and check the data compared to SARS-1 or other or MERS or other coronavirus. Why there are so high percentage of neurotoxicity or even the emotional change happen? Why the percentage of the blood system problem like clot, immune disorder happen in SARS-CoV-2? So you should assume this related to the functional change. And before we really understand what happened in the spike protein, this kind of injection with the vaccine, no matter what vector or what technology, always potential risk for us. And the other thing is, my last message is, I really hope that all the doctor can tell the things you understand about the COVID from the meeting or from other doctors to your patients. We face a lot of stress, but we should know if we don't do it, we doctors are always in the front lines to get hurt from this kind of disaster. And also, I hope also doctors can work together that we can make some, maybe the new system to record the adverse effect, which is better than the CDC or NIH's one. And also we can collect the data from our patients and work with the other doctor who has the medical labs like Dr. Ko and Dr. Urso. And also we can make our system, because we are doctors, we work with patients, so we have the first-hand knowledge. Why do we sit there, wait for some people only stay in the lab and the office to tell you what to do? I stay in the research lab. It's the top coronavirus lab, but let me tell you, without doctors' experience, those top scientists, many of them, they don't have MD degree. They can even not interpret the data connected to the clinical symptom. They just sit there, say the data happened from the, the petri dish, or happened in the mouse, and oh, this is what I want to know. But totally, there is a big gap, lose the connection with the clinical results. So doctors have, in this whole pandemic, doctors are suppressed. So it's unlike mid-age religion. Now we doctors should use the data and our network to work together and collaborate with the medical labs. They want to do something to help people. They want to deliver the truth. We all work together. This will be more powerful than those research labs or WHO or all these kind of authorities.
Thank you. Thanks to our very talented panel. Thank you for being here. And we'll have more time for more questions tomorrow during Dr. McCullough's session. Thank you. Enjoy your dinner.
Yeah, and dinner is, I believe, ready out there. We're not supposed to really start dinner for another 8 minutes, but if for those of you who would like to be first in line, go ahead. Otherwise, take a short break and then grab some food. I think our aim is to start the dinner program at 7, so yeah, we're getting the bang for your buck today. Yeah. Yes. Yeah. No, I will. Yeah, I can get you a thumb drive. Okay. Yeah. Yep. You'll get it. Okay. I can't stop on my face. Thank you, Dr. Powell.
We met. By all means, thank you for asking. Okay.